Good day, ladies and gentlemen. I want to thank you for inviting me to give this talk. I hope that you will enjoy it and that it will illuminate some of the interesting facts about ticks and why they are such good models for biology. Now, before I jump into the main topic of the talk, let me just give you some background on myself. I did my PhD degree at the University of Pretoria in South Africa in the laboratory of Albert Knights, working on ticks and salivary gland proteins. I then moved to the laboratory of Eugene Koonin at the National Center for Biotechnology Information, where we worked on the nuclear pore complex and the nuclear envelope proteins and how it relates to eukaryotic evolution. That was a lot of bioinformatics work, so I longed for the lab again. And a year later, I moved to the laboratory of malaria and vector research under the mentorship of Jose Ribeiro. And we worked there on various blood-feeding vectors, such as mosquitoes, ticks, and assassin bugs, where we looked at protein function, protein structure, as well as protein or salivary gland transcriptomics. I moved back to South Africa in 2008 and I joined the Agricultural Research Council at an institute known as Onderstepoort Veterinary Research as a senior researcher. And I'm still there. I am now a principal researcher and a research team manager. And we work on a variety of agricultural problems that includes diagnostics, insects of veterinary importance, as well as ticks. So just a brief background on the Agricultural Research Council. We are an institution with roughly 11 institutes scattered across South Africa, where we perform a variety of different research agendas, topics on many different agricultural problems. So the institute that I'm located at, Onderstepoort, actually perform diagnostics for controlled diseases, controlled animal diseases, such as, for example, foot and mouth disease, East Coast fever, African horse sickness, blue tongue virus, Rift Valley fever virus. And we also house the Gertrude Tyler Tick Museum. And we also produce vaccines for a variety of diseases. So to get to the main topic, why are ticks the biologist's dream model? I just want to indicate that biology as a discipline has now been in existence for several hundred years. And within that discipline, a number of sub-disciplines has arisen. And these, in fact, have matured into their own disciplines in their own right. Things like taxonomy and systematics, biochemistry, genetics, microbiology, zoology, medical and veterinary medicine, epidemiology, molecular biology, genomics, proteomics, transcriptomics, physiology, ecology. So all of these can be studied when you look at ticks. And in fact, I'll show some examples where people uses most of these different sub-disciplines to actually study ticks. And that is why I claim that ticks are a biologist's dream model, because they're really amenable to a lot of these sub-disciplines and they can give you a lot of information regarding these sub-disciplines. Why are ticks important? Well, if you live in the USA, you will know that Lyme disease are a well-known disease. Almost everybody knows about it. You find it in the news, celebrities reporting that they are actually affected by it. And the CDC reporting at least 300,000 cases a year. And every year, the number of people infected is increasing. If you do a search with the terms ticks and disease, and you obtain the abstracts of that search, and you analyze those abstracts and their titles, and you construct what we call a word cloud. Then you can see that this word cloud contains various terms of different sizes. Those different sizes are important because it indicates to you the number of times a certain term appeared within that search. In this case, we obtained 8,427 records. You can see a breakdown of these records, show you tick vectors, Ixodus ricinus and Ixodus scapularis. These are in fact the vectors in Europe and in the USA. For Borrelia burgdorferi, the causal agent of Lyme disease. You also find for diseases Lyme disease, spotted fever, tick-borne encephalitis, and for pathogens anaplasma, phagocytophilum, relapsing fever, rickettsia, spirochete, and for host cattle, dog, human, livestock, rodent, and mice. Now, if you look at this word cloud, you may think, well, that's what ticks is about. The reality, though, is that ticks affect many more animals across the globe than probably humans. And therefore, ticks are at the center also of what is known as a One Health perspective. Now, the One Health perspective presents the concept that animals and humans and the environment actually interact in a holistic whole, so that the health of animals also affect the health of humans, and the health of the ecosystem will affect both animals and humans. And therefore, you have to look at everything if you want to consider human well-being. Ticks in this case play a central role because ticks transmit diseases to both animals and to humans. And here's a few examples. Ticks can transmit bacterial diseases such as heartwater, such as tick bite fever, such as relapsing fever. Ticks can also transmit protozoan organisms that causes diseases such as redwater, babesiosis, East Coast fever, theileriosis. And ticks can also transmit viruses such as Crimean Congo hemorrhagic fever. And not only that, but ticks can actually also affect hosts directly by causing, in some cases, paralysis. Now, this realization that ticks are part of the One Health paradigm actually came forth a number of years ago when we had the last Ticks and Tick-borne Pathogens conference held in Cairns in Australia, where the whole topic of that conference was One Health. So it sort of indicates to you the realization in the field that ticks play an important role in both animal and human health. The next thing that sort of come out of the One Health perspective is that ticks are geographically distributed across the globe, and you find ticks in various different ecozones or ecological habitats, where they are restricted due to the fact that they evolved there due to speciation. They are restricted to geographic habitat because their host range is restricted. They are limited by the habitat itself. However, over the last few years, it has become evident that some ticks do not recognize or respect borders, and due to the movement of animals and humans, some tick species are rapidly distributing across the globe and entering countries where they are not wanted or needed. So this has led to a very strong and healthy discipline where people model tick distributions, linking with their ecological habitats and their habitat suitability, and where they try to predict where ticks will be distributed perhaps in 50 years' time, and by using that, predict the type of risks that we may face with regard to emerging diseases. So if you're interested in modeling, if you are an ecologist, if you like computer work, but also like maps, then tick geographic distribution is definitely something to consider. It is and remains a very interesting subject. Another area where people that work in this field sometimes engage in is diagnostics of tick-borne diseases, and that is because obviously you have to identify the pathogens to treat animals or humans, and identify perhaps sometimes even the ticks. So many laboratories across the globe are involved in diagnostics, especially where the movement of animals is concerned. This is in many cases controlled by governments. And the people that work in these labs, they're involved in test development and the validation of those tests. So test needs to be specific and sensitive to be utilized. And many people actually wants to develop the most specific and the most sensitive tests. To do this, you need to understand the epidemiology of a disease. You need to understand the genetic diversity of pathogens that circulate within a specific host. You need to also understand the genetic diversity of non-pathogen organisms that may affect your test that is also circulating within a specific host. And you have to accredit these tests. So a lot of research then goes into understanding biodiversity of pathogens in ticks and in their hosts, so that you can actually understand whether a test will be specific or not, whether you're actually detecting the right pathogen or not. People working within this field, diagnosticians, actually have a direct impact on disease control on a daily basis because they test real samples that come in from clients. If you like to make a direct contribution to society, then diagnostics is definitely a space where you can work in. If we go to a bit more basic aspect, out of the work that was done and performed on pathogens in ticks, the realization came about that ticks do not only harbor pathogens, they actually also harbor non-pathogenic endosymbionts. One of the questions then is, why do ticks have these microbial communities in them? And one reason is that, well, ticks cannot make their own vitamins, so for some of these, they actually need this commensal relationship with the bacteria to survive. And what people are these days looking at, they're looking at microbial communities as networks of bacteria that are all contributing, and that it's the bacteria or the microorganisms in the tick, it's the microorganisms in the host or on the host, inside the host, and they all interact in various ways. People are interested in how these communities interact with each other. So if you are a microbiologist or you have a leaning towards microbiology, then this is a very vibrant field within the tick community itself, where people are doing a lot of interesting work, and they're actually using a lot of cutting-edge technologies to understand how these microbial communities interact. Some people want to control ticks and the pathogens. These people work in laboratories where they either try to develop new vaccines. These vaccines could be live attenuated vaccines, they could be inactivated vaccines, they could be subunit vaccines. And for this, you probably sometimes need to sequence the pathogen genomes, you need to express proteins and test those vaccines. So a lot of diverse techniques is actually needed to develop a good vaccine. Some people focus on plants because it is known that some plants will repel ticks. Some plants may contain substances that will actually kill ticks. So some people are looking into phytomedicine to develop the new range of acaricides that will be used in the future. Then some other people also working on vaccines, but in this case, not just working on a vaccine against the bacteria inside the host. They're actually trying to prevent the pathogen from entering or infecting the tick. So they may focus on gut membrane proteins that may be receptors of the pathogen. They may focus on salivary gland receptors that may also be the receptor of the pathogen. And they're trying to develop transmission blocking vaccines. So a vaccine that is made in the animal, but when the tick ingests the blood, it also ingests antibodies. And those antibodies will then block infection of the tick by the pathogen. So also a very interesting field that is on its way to perhaps deliver some results in the future. if you're not very lab-based, there are people that actually are trying to prevent the importation or the introduction of ticks into their countries of origin. These people work in customs, they may work in government, and for them it's quite important that they can actually understand and identify ticks, the right ticks, foreign ticks. For that, you need to be a very knowledgeable entomologist or a acarologist. And so you can say that if you cannot identify the tick, you cannot control the risk. So a lot of people, if you like to identify things, you even find people riding out trying to find cattle wandering around over the borders, capture them, and you will work closely with people like these. This is a much more hands-on type of experience than perhaps working in a laboratory. So from this, if you cannot identify the tick, then you cannot control the risk, comes one of the first basic subdisciplines that we can look at, and that is the field of classification and taxonomy, systematics. Where taxonomy is the branch of science concerned with classification, especially of organisms. Another way to describe taxonomy may be the description of families, genera, and species, while systematics is the study of the evolutionary relationships among organisms. Now you can see down here below, I show you 5 different ticks. They're supposed to be 5 different tick families. 3 of these exist in the world today, the Mite-y, the Argasidae or the soft ticks, and the Ixodidae or the hard ticks. Now, 2 families is extinct. You only find them as fossils in amber, the Chimeridae and the Dinocrotonidae. Chimeridae was just recently described. Very exciting new family because this tick shares characters between hard and soft ticks. If we look at tick taxonomy and the description of species over time, you will see that tick species actually, the first tick species were described around 1758. And over the years, as with most genera or families, there was an increase in the species numbers. Currently, there's roughly 1,000 ticks described, about perhaps 800 hard ticks, 200 soft ticks. What I want to draw your attention to is the year 2000, and you can see after the year 2000, there's still a very smart increase and a sharp increase in the discovery of both hard and soft ticks. Now, this is important because it tells you that tick discovery is not over. Okay, my daughter once told me that what will she discover one day because everything has been discovered, and sometimes in taxonomy, you may feel the same way because ticks are a very small group and they certainly all been discovered by now. The reality though is that as we look for ticks in strange places, new places, people always find new tick species. And I believe this trend will continue for the foreseeable future, especially because now we have introduced molecular biology techniques that even gives us a better perspective on what actually happens within ticks. What about tick systematics? What is the place of ticks in the world? So we know that ticks are eukaryotes. We know they're molting animals, they're arthropods, they're what is known as the Chelicerata. They are arachnids, which they share with spiders and scorpions, and they're part of what we call the acari, the mites. Within the mites, they're part of what is called the Parasitiformes, and they share this distinction with 3 other main lineages, Paleoacarida, Holothuria, and the Mesostigmata, and then the ticks are broken up into the various families and genera. If we look at tick systematics on a family level, though, some interesting things emerge. So one of the first questions that you can ask is, what is the relationships or the relationship between the different families? And the answer to that is not so simple. So for example, Mite-y, which shares characters with both soft ticks and hard ticks, you can see it's got a sort of leathery integument like a soft tick, but it's got a seroscutum almost like a hard tick. and you can sort of see some of its mouth parts from the top, like a hard tick. You cannot see it for soft tick. So there's some features or traits or characters that is shared within this tick. If you do molecular biology analysis, a number of studies have found different things. Some studies have found that the Mite-y shows a sister relationship to the Holothuria. Some studies show the sister relationship to the hard ticks. Some studies show the sister relationship to the soft ticks. And some studies indicate that it is a basal lineage. Now, resolution of these relationships is very important since it will have profound implications for the evolution of blood-feeding behavior. And what I can tell you is, while this is my favorite option, it's not by a long shot defined or over that this will be the final answer to the question. So there's a lot of scope to make some interesting discoveries regarding the relationships of these families. And then obviously some new questions that enter is, where would these old extinct fossil families sit in the tree? So a lot of work that can still be done, that will be done, and that will remain interesting. Why is this also important, these relationships between the families? And I can show you two examples. If you look at hard and soft ticks, you actually find an interesting observation that was made many years ago. Soft ticks have a leathery integument that rapidly expand within minutes of feeding. Hard ticks, on the other hand, go from this to this over several weeks, several days or several weeks. Soft ticks can feed multiple times fast, and hard ticks only slowly imbibe over a few days, and they only feed once. Soft ticks can lay several egg batches, while hard ticks only lay one egg batch. And perhaps to me, one of the most interesting ones, soft ticks secrete excess water from the blood milk via the coxal organs, while hard ticks secrete it via the salivary glands. So based on this, we have speculated before that they have different life history responses to the same environment. And we asked the question is, whether this indicate independent evolution of blood-feeding behavior, even though systematics indicate a monophyletic origin for the two families. If you look into the ticks themselves and you look at processes that regulate the host defenses, so you will recall that when a tick feeds, it needs to secrete many different proteins into the feeding site to inhibit the host defenses. What you find then is that hard and soft ticks will actually target the same targets, but they will use different molecules to do so. And again, the question is raised, is this indicative of independent evolution of anti-hemostatic and immunomodulatory mechanisms, even though systematics indicate a monophyletic origin? So again, by far not yet resolved. We have some ideas on why this is like this, but definitely a really interesting sort of mystery in tick biology that still needs to be resolved, and perhaps you can do it. Let's return to tick systematics, and we look at hard ticks at the genus level. And you will see that although the first tick was described around 1758, it was only around 1795 that the first tick genera were raised. And what you then see is, and it was this Ixodidae ricinus, the tick that was also first described. What you then see for hard ticks is that over time, the list of genera increased as people were discovering more ticks and describing them. And this increase stayed relatively stable. It's only in the year 2000 that we see something happening here with these red boxes. So in the first instance, the genus Aponoma was sunk, and members of the Aponoma were either shuttled to the genus Amblyoma, or they were shuttled to a new genus that was created called Bothriochroton. Similarly, the genus Buophilus was sunk, and all members of Buophilus were divided or were actually placed within the Rhipicephalus. Quite recently, 2 new hard tick genera were also raised, Archaeocruton and Ruberticus. Both of these were previously known as Amblyomma as well. And you may ask the question, why did these changes happen? One of the reasons is that around 2000, you saw the rise in extensive application of molecular biology techniques for ticks. And with the advent of molecular biology applied to tick systematics, we saw that the molecular biology indicates to us that many of the lineages were paraphyletic, they were artificial genera, and therefore they were broken up into lineages that may resemble the evolutionary history of the ticks better. We move to the soft ticks, it's perhaps even more interesting because it may be more illuminating. So for soft ticks, the first genera was described along the same time as the hard ticks. But something interesting happened here. Soft ticks were kept to the minimum of genera. So from 1800 to 1850, you effectively find only one genus called Argas. From around the 1850s to 1900, you find Argas and Ornithodorus. From 1900 to 1950, you find Argas, Antricula, Ornithodorus, and Octobius. Then from 1950 to the year 2000, give or take, or basically up to 2019, you find Argas, Antricula, Notoaspis, Ornithodorus, and Octobius. So very fewer genera than you see for hard ticks, even though many different subgenera were created over the years to describe various ticks that had very different morphologies. So quite recently, we proposed that the number of these subgenera needs to be raised to genus level. So now there's many more genera that actually exist, and we have the feeling, based on already on the molecular data again, that we may have to raise some more genera in the future again. The reasons for this is that the molecular data simply indicate that within soft ticks, there's many independent evolutionary lineages. This may be indicative of a long time ago when many of these genera actually originated. Another field that is sort of linked to taxonomy and systematics is that of paleobiology. And this for ticks is quite interesting. So you do not find ticks within rocks, but you do find them as fossils in amber inclusions. And what's interesting about this is that you find a variety of ticks that are actually extinct. So at the top here, we have 4 extinct genera: Dinocruton, Chimera, Complurischitula, and Cornupalpatum. They were all found in Burmese amber dated 100 million years ago. What's perhaps even more interesting is that within the same Burmese amber, we actually find tick lineages that live today. Things like Amblyomma, Hamaphysalis, and Ixodidae. You also find soft ticks in amber, and this tick was found in a different amber, New Jersey amber, but dated almost to the same age as the hard ticks. Now, why is this important? These ages gives a minimum date of origin for ticks, and you can use these ages to actually calibrate various nodes on a molecular phylogenetic tree and use that for molecular clock analysis. What we end up with is a tree where you can see there's the 100 million year cutoff for the fossils, with the implication that ticks are much older than 100 million years. And our current estimate is around 250 to 300 million years ago. So it's quite important. It gives us some insight on where ticks may have originated. Now, again, few tick fossils have been found so far. So as we collect more data, this picture may change. So again, a very exciting time to be in tick taxonomy and systematics. tick fossils can also be fun. Tick fossils are nice for tomography, so you can create beautiful digital images of ticks that's fossilized, and you can even make a movie, and you can look at these ticks. And my prediction is that in future, tomography will be used more and more for live or extant specimens as well, meaning that perhaps somewhere in the future, we could have a vast database of digitized tick species that anybody can look at any time, and actually with high resolution, where you can look at morphological features that may be used in future research. So I believe a very exciting time awaits people that work in this field. What type of techniques will you use if you are a taxonomist or you're working in systematics? So in the first instance, you do a lot of light microscopy, and that is quite interesting because you can see the ticks. You will also learn many different terms, arcane terms, in fact, that nobody else will know except you and a few other select acarologists. You can perhaps excite people at cocktail parties by reciting these, although I must say that is not always a recipe for success. Things like the femur, the tibia, the metatarsus, the hypostome, the basis capitulum, the coxae, the genital apron. So all terms that you can learn that describe the morphology of ticks. Moving to scanning electron microscopy, you can take high resolution images of larvae, of nymphs and of adults. That is beautiful under electron microscope. It's a good technique to know. You can also perform morphometric analysis with these features, where you look at the shape or some landmark points that describe a specific morphology, and you can compare them within a species or between species. And this can actually tell you whether some closely related species are true or not. Or you may be looking at one species complex and analyzing it, and then through time you realize that you're actually sitting with more than one species because the morphometrics are telling you that you actually have more than one species in your sample. So quite important and exciting technologies that are now being developed that you can take very far with you. You can also perform molecular analysis, phylogenetic analysis. You can do this on single genes, on multiple genes, on whole mitochondrial genomes, or even on whole genomes. So this is a very exciting field where a lot of development and a lot of work is taking place at the moment, and definitely for the foreseeable future. So my take on systematics and taxonomy is that it will definitely be a vibrant field for many, many years to come. If you're interested in more talks on tick systematics, you can actually visit the Tick Mitochondrial Network on YouTube, where there's a number of presentations on a variety of topics within the mitochondrial genomics field, and where you can learn more about this field if you're interested. Let's move to another field, tick physiology. Now, ticks have to do a number of things if they want to get a blood meal. They have to detect the host. They have to attach to the host. They have to interact with the host and actually overcome the host defenses. And they have to do something with the blood meal. They have to process the blood meal, digest it, and they have to get rid of water that is present in the blood meal. And the big question for me, always of interest, is how much or how many of these characters involved in this is conserved in the ancestral lineage to ticks. It tells us something about evolution of blood feeding behavior. So let's first look at host detection. So ticks have a little organ known as Haller's organ that sits here on the first leg of the tick. And if you walk in nature, you may find ticks sitting on a leaf or on grass, and you'll see them extending their feet, waving it around. They are trying to smell you by using their olfactory organ called Haller's organ. If we look at Haller's organ, it's quite interesting. There you can see a little tick waving again. First, it was Haller himself thought it was an auditory organ. Batelli, after him 10 years later, discovered it was used for questing, and he described his questing behavior. And then Lahil actually indicated that it was an olfactory organ. And the realization came through time that all ticks have this organ. Some mites also have this organ, so it's not tick specific, but it's definitely an organ used for sensing. One of the big questions is, how does ticks actually sense with this? So the general idea or model is that there's receptors in these organs, and the volatiles can come in and they bind perhaps to some odorant binding proteins that then can take the odorants to a receptor, and then obviously there's some signal transduction events that tell the tick what it is smelling. Now, to understand this, people have done different things. They've actually tried to determine using something known as proteomics, detecting the proteins and characterizing both Haller's organ and some other legs that do not have Haller's organ. And what they found were interesting. They find that soluble proteins known to be able to bind odorants, lipocalins is one family that was present. Niemann-Pick type 2 proteins is another family that was present. But strangely enough, odorant binding proteins and chemosensory proteins that you actually find in insects were not present. This tells us that ticks may have a completely different mechanism of smelling things. People that performed transcriptomics on these 4 legs also discovered ionotropic receptors and gustatory receptors. And so one of the big questions currently is regarding the detection of carbon dioxide. So both soft ticks and hard ticks can detect carbon dioxide, and insects detect carbon dioxide via the GR2 receptor. And what you actually see is that this GR2 receptor is absent in a variety of insects, but also in ticks. So one of the main questions now is, will this carbon dioxide receptor be an ionotropic or gustatory receptor, or perhaps something completely new? So that will be interesting to discover. If this receptor is something new, it will again indicate that most probably ticks evolve blood feeding behavior independently from any other lineage. We move to the next stage. So the way ticks attach. Ticks attach using what is called the hypostome. The hypostome, you see the hypostome here, it's also comprised out of chelicerae. These are these cutting organs. What ticks do with this, with their mouth parts, is they cut with the chelicerae, and then with a ratcheting movement, they actually pull the tick hypostome into the feeding site, and they anchor the tick there. And what we know is that this hypostome is a very unique character to ticks, but it's found in all ticks. It may differ in form and shape and size, but all ticks have it. So probably, again, it's indicative of the fact that ticks evolved a blood feeding lifestyle in the ancestral tick lineage. So linked to this, when the tick attaches, the next thing is to engage with the host and to inhibit all these different defense mechanisms that the host have. They have molecules that target pain responses, so that when the tick bites you, you do not feel it. They secrete molecules that actually prepare the feeding site for engorgement. So they create a cavity from which they feed. They are pool feeders. So they create a hematoma. And then they target many other host-derived things such as blood clotting, platelet aggregation, vasoconstriction, the complement cascade, and vascular permeability. So ticks need to do all these things, and they do this by secreting a few hundred to a few thousand proteins during feeding. And one of the big ways to study these proteins and to actually understand what they do is to look at the salivary glands, because these proteins derive from these salivary glands that you find in the tick. These salivary glands are really interesting. They're beautiful organs. They look like little packets of grapes. What you see here, these little balls are called acini. So it's constructed out of these acini. And if you look within an acini, you will see that the acini itself is constructed out of multiple cellular types. And those cellular types, in this case for soft tick, is packed chock and block full of protein granules that is secreted into the host when the tick bites you. One of the challenges that we have is we want to understand how many of these proteins there are that's secreted into you and what do they actually do. So to do this, you use a number of analytical techniques. So if you are a biochemist, then the study of salivary gland protein biology is quite an exciting field. Okay, because for two reasons. Proteins are abundant in the salivary glands, and secondly, the proteins are soluble because they're secreted, and therefore the proteins are quite stable. This makes them very nice proteins to work with if you're somebody doing biochemistry on them. So you can purify the proteins using chromatography. You can analyze them using gel electrophoresis. You can sequence their N-terminal sequences. You can determine their intact masses using mass spectrometry. You can characterize their functions and titrate their functions out and get to some functional parameters using functional assays. You can determine their structures and determine their structure, function, and mechanism relationship. You can run more gels and do proteomics on the spots and identify proteins. You can sequence their transcriptomes and analyze them, looking for differential expression, classifying protein families, performing phylogenetic analysis, and effectively a very satisfying type of endeavor if you are more leaning towards the analytical laboratory type of person. So very exciting and a very nice model because it's easy to work with. And because there's so many functions that has not yet been discovered, the options to make some nice discovery is very good. So this is definitely something that you can look into if you're an analytical biochemist. So why do we do all these studies, and why do we want to understand what is going on at the tick feeding interface, and why ticks can actually be successful in obtaining a blood meal? And the answers to that is fairly applied. Using the knowledge, using the identity of the proteins that we identify, we can actually go and select some of them and test them for their potential as vaccines. So one of the reasons is to develop anti-tick vaccines. The other reason is that you can actually take these proteins that you get from the tick and you can develop new therapeutics using human or animal medicine. And the idea behind this is that ticks has evolved these host defense or anti-defense mechanisms over millennia. So many millions of years, ticks have adapted to vertebrates, have overcome their defense mechanisms. They are optimal pharmacologists. Therefore, you don't go out there and try and design the new drug yourself. You can go to the ticks and let them design it for you. You characterize it, you find the functions, and after that, you may be able to optimize it, making a new medicine. So two very applied reasons why we actually do this work, beyond the fact that sometimes it's just nice to understand the complexity of what is going on at the feeding site, because that alone is also very interesting. And there's mysteries here for each of these topics. You can probably design a research agenda and spend many years on uncovering what is going on in each of these sections of this chart. If you don't like analytical biochemistry, but you do like physiology, you do like to work with things that is more on perhaps an organismal scale, then you can look at things like salivary gland secretion, the innervation of organs in the tick. For example, here's a beautiful study where the innervation of the salivary glands were elucidated, and where you can see these nerves actually derive from the central nervous system of the tick. So the tick also has a central nervous system, and if you can delineate these nerve patterns, you can actually learn there. You can see those nerves going into the little acini. You can actually discover how the tick knows when and where to secrete the protein content that is stored within the salivary gland. So a very satisfying field of study. Nice thing there is ticks have many, many different organs beyond the salivary gland. That's also innovated and that also function with some receptor hormone regulation. So if you're a physiologist, ticks have a lot in store. And just to show you here, here we have some physiology in action. I want to show you here what we observed in real time.
Yeah.
Bio.
That's bio.
You can see there, this tick actually secreting water via the anal pore.
Zie je?
This was quite an interesting discovery when we were doing this, because the Nathalilidae secretes water completely different from what you see with either the hard or the soft ticks. So, and I believe personally that working on these ticks, a lot of new physiology is still waiting to be discovered. We go to the last sort of stage that we identified in tick feeding, that is blood meal processing. A lot of good work has been done over the years on blood meal processing. So, for example, the first hemoglobin crystals were observed in the gut lumen in 1861. In 1934, Roosler actually showed that ticks were digesting their food intracellularly within the cells of the gut. And this is quite interesting because it's different from what you see in insects or perhaps other insects where digestion of any meal is done extracellularly. So ticks actually take up the blood and they digest it intracellularly. And this was done almost 100 years ago. 1979, Balashov actually showed that the uptake of food is receptor mediated or is mediated by receptor mediated endocytosis. And a few years later, Lara et al. Showed that hemosomes form. Hemosomes are very important. They are structures that allow the tick to detoxify and deal with these large amounts of heme that is generated or that's present in the blood meal due to the fact that hemoglobin has a heme molecule. Moving to a molecular level, a lot of good work was done in the laboratory of Peter Kopasek and his colleagues, where they have actually shown and discovered how hemoglobin and albumin, main proteins involved in the blood meal or present, is broken down using a cascade of different digestive enzymes. So they've shown that it's first chopped up into large fragments and then smaller fragments until you end up with dipeptides. This may have been expected, but to actually confirm it and to show that this is what happens is what is really exciting. And we know that most of these digestive enzymes is in fact present in all ticks, with the implication that blood meal digestion, intracellular blood meal digestion, probably evolved again within the ancestral tick lineage, or perhaps even in some of the associated mite lineages. So this is the end of sort of the laboratory-based work that you can do with ticks. So these are for people that like technology a lot, for people that likes to be at the cutting edge of new innovations, where they can combine laboratory work with bioinformatics, with computer work, and where they can work with molecules. What if you are not one of these people? What if you want to do something else? Is there scope for you? And what I can mention is that whether you work in the lab with a tick, whether you work with the modeling of tick distribution, someone somewhere have to collect these ticks for you. So to construct this specific picture meant that each and every dot that you can see here is a coordinate where a specific specimen were collected and were identified to be the tick Boophilus decoloratus, today Rhipicephalus decoloratus. So there's people out there that go out and understand the ecology of a tick and understand how to collect ticks. And I can tell you that this part of a biologist's life, of a tick biologist's life, is sometimes the most rewarding. Because you're out there in nature, you can see different habitats, you can engage with ticks and with their relatives, and you can see where ticks actually live. So if you want to be out there and collect new ticks, then tick biology also has a place for you. In fact, I can refer you to a nice video by Sebastian Munoz Leal, who presented a very nice talk on the tick genome network that deals with tick collection in the field. And it's a very entertaining talk. It's one I enjoy quite a lot, so I encourage you to actually visit it and look at it. The other nice thing is that collection of ticks can take you to places that you simply did not think of that you'll end up with. For example, climbing a mountain, climbing a hill, crawling into caves, looking under rocks, all places where you won't necessarily expect to find ticks, but where you do. So why do we do all this work in the end? Laboratory work, tick collection, geographic distribution. Well, in the end, you have to put it all together and take the systematics, take the phylogeography, look at the morphological adaptations, the physiological adaptations, what happens at the molecular level, put it all together into a whole so that we can actually understand how ticks evolve the blood-feeding lifestyle. That is what I'm interested in personally, because you can actually look at the whole of tick biology and try and understand it from this perspective. As Theodosius Dobzhansky said, nothing in biology makes sense except in the light of evolution. And I want to paraphrase him, nothing in evolution makes sense except in the light of ancestral reconstruction, reconstructing the events that happened before different lineages diverged. I want to end off with my wall of tick friends. These are people long gone, still existing, but they all had an impact on my development as a tick biologist. I got ideas from them, I got critique from them, I got good arguments from them, but all of them contributed significantly to my own development as a tick biologist, as a biochemist, as a parasitologist. So I want to thank them all. Also, many of the different funders from which I have obtained funding in the past. Thank you. I will now take questions.